Welcome to Biotanemy.
Tricarboxylic acid cycle. TCA cycle or tricarboxylic acid cycle, known as the Krebs cycle, after Hans Adolf Krebs, the scientist who discovered the pathway in 1937, and received the Nobel Prize in 1953 for physiology or medicine. Also, called the citric acid cycle since the first stable compound formed in the pathway is citric acid or citrate. And citric acid contains 3 carboxyl groups. Hence the name, tricarboxylic acid cycle. It is the central hub of cellular respiration where carbohydrates, fats, and proteins are oxidized to generate energy in the form of NADH, FADH2, GTP, ATP, as well as carbon dioxide as a byproduct. These reduced coenzymes will later donate electrons to the electron transport chain, ETC, to produce large amounts of ATP. The cycle occurs in the mitochondrial matrix of eukaryotic cells and in the cytoplasm of prokaryotes. During the TCA cycle, acetyl groups are completely converted into carbon dioxide and reducing equivalents, as well as providing intermediates for amino acid synthesis, fatty acid synthesis, heme synthesis, and gluconeogenesis. Thus, the cycle is amphibolic since it gives rise to both catabolic and anabolic compounds. We'll see it in later part of video. The cycle consists of 8 enzymatic reactions. Step 1 is the formation of citrate. The enzyme citrate synthase will condense oxaloacetate, a 4-carbon molecule with acetyl-CoA, a 2-carbon molecule to form citrate, the 6-carbon molecule and release CoSH, which is an irreversible reaction, and the step is highly regulated. Step 2, formation of isocitrate, catalyzed by a conatase. In this step the citrate is rearranged into isocitrate through dehydration and rehydration. During this process an intermediate cis-aconatate is formed. Step 3, oxidative decarboxylation of isocitrate. The enzyme isocitrate dehydrogenase will remove 1 carbon from isocitrate in the form of carbon dioxide to form alpha-ketoglutarate and NADH. This step is activated by ADP and calcium, while inhibited by ATP and NADH. It is the first oxidative decarboxylation in TCA cycle. Step 4, a formation of succinyl-CoA is catalyzed by alpha-ketoglutarate dehydrogenase complex, and is the second oxidative decarboxylation to release carbon dioxide and NADH. The cofactors required for this reaction are thiamine pyrophosphate, lipoic acid, FAD, NAD+, and CoA. And the process can be inhibited by arsenite poisoning. Step 5, a formation of succinate, which is catalyzed by succinyl-CoA synthetase and generates GTP. This step is known as substrate-level phosphorylation. Step 6, a formation of fumarate, catalyzed by succinate dehydrogenase. It is the only TCA enzyme embedded in the inner mitochondrial membrane and also function as ETC complex 2. Step 7, formation of malate. The enzyme fumarase will add water across the double bond of fumarate to form malate. Step 8, regeneration of oxaloacetate. Finally, the malate is converted to oxaloacetate by the enzyme malate dehydrogenase to complete the cycle. Now, the net reaction of the TCA cycle, it takes acetyl-CoA, 3-NAD+, FAD, GDP, inorganic phosphate, along with 2 water molecules to give 2 carbon dioxide, 3-NADH, 1-FADH2, GTP, CoASH and 3 hydrogens. The ATP yield from 1-acetyl-CoA is calculated as 1-NADH will give 2.5 ATP. So 3-NADH will give 7.5 ATP. 1-FADH2 gives 1.5 ATP. And 1 GTP is equivalent to 1 ATP. Hence 7.5 plus 1.5 plus 1 is equal to 10. And if we consider that acetyl-CoA comes from the glycolysis cycle, and 1 glucose can form 2 acetyl-CoA, then 2 times 10 is equal to 20. Hence, 20 ATP are created via 1 TCA cycle. Now, the catabolic role of the TCA cycle is to break down acetyl-CoA. While speaking about the anabolic role, it provides intermediates for other pathways like 1. Citrate will take part in fatty acid synthesis. 2. Alpha-ketoglutarate in amino acid synthesis. 3. Succinyl-CoA for the synthesis of heme. And 4. Oxaloacetate is required for gluconeogenesis and aspartate production. The anaplerotic reactions catalyzed by pyruvate carboxylase will supply oxaloacetate to replenish TCA intermediates by converting pyruvate, carbon dioxide, with the help of biotin as cofactor and expending 1 ATP to produce oxaloacetate, ADP, and 1 inorganic phosphate.
That's all for today's video. Thank you for listening. Hope all of you understand the concept, and for more queries and proposal, please comment down. See you all in a new topic.